Bueno, pues muy buenos días a todos. Hoy tenemos un seminario más de la serie de Frontiers in Genomics. Como les anuncié en nuestro correo publicitando el seminario del doctor Corces, este va a ser el último seminario de este semestre escolar de la UNAM y reiniciaremos el 1 de marzo. Ahora es para mí un gran placer realmente introducir al doctor Corces, que va a dar el seminario de hoy. El doctor Corces es, como pueden ustedes ver, me va a perdonar, pero es muy joven y tiene realmente, esos son los privilegios de alguien que ya es mucho mayor. Y el doctor Corces recién fue contratado por los institutos Gladstone de la Universidad de California en San Francisco, y son el Gladstone Institute for Neurological Disease y el Gladstone Institute for Data Science and Biotechnology. Él es egresado de la Universidad de Princeton, donde estudió biología molecular y ciencias de la computación, y después procedió a hacer un estudio de posgrado en biología del cáncer en Stanford, y también en epigenética, y también hizo su postdoctorado ahí mismo. El doctor Corces realmente es un experto en el manejo de datos, de grandes bases de datos, y también en el manejo de datos de biología molecular, y ha sabido justamente conjuntar esas dos disciplinas de una manera muy exitosa, y puede realmente con eso abrirse el camino para entender muchas de las enfermedades tan complejas que ahora estamos descubriendo las bases moleculares, pero justamente utilizando las dos herramientas que él maneja con tanta dexteridad, que es la biología molecular y la epigenética, y la biología y el manejo de grandes bases de datos. Entonces, dicho esto, pues le quiero agradecer el haber aceptado participar en nuestra serie de seminarios de Frontiers in Genomics, y de haber participado también en la clase con los estudiantes de la licenciatura. Ryan, thank you very much, and again, welcome.
Thank you. Thank you all for joining. It's really a great pleasure, and I look forward to telling you a little about the work that's going on in my very young and new lab over here at Gladstone Institutes and UCSF. So my work is predominantly focused on understanding this puzzle of the nucleus, and in particular, we're interested in neurological diseases and how epigenetics contributes to these various neurodegenerative disorders, primarily including Alzheimer's disease and Parkinson's disease. So just starting from kind of the base fundamentals of what epigenetics is and how the epigenome is co-opted, we understand that enhancers talk to promoters, and that this forms the basis of much of the transcription that occurs within each cell. And this process is controlled by the binding of sequence-specific transcription factors, which can interact with enhancers and promoters. The binding of those transcription factors and the overall state of these enhancers and promoters is controlled by DNA methylation in many cases. And of course, we know that this process doesn't occur in isolation on this linear piece of DNA, but rather the DNA folds in 3 dimensions due to activity of proteins such as CTCF and the cohesin complex. And all of this quite intricate epigenetic machinery can be regulated or dysregulated by changes to histone tail modifications or wholesale nucleosome remodeling by the BAF complex. What got me interested in this intricate epigenetic process was I was studying cancer initially in my PhD, and all of these factors were coming up as mutated in cancer, as kind of cancer genome sequencing was taking off. But what's maintained my interest in the epigenome over the years is that you could use this slide for essentially any disease, And so now I'm interested in the brain, and all of these epigenetic factors are regulated or dysregulated in a variety of neurological contexts, including learning and memory or neurodevelopmental disorders. And so I view this as a really interesting way to understand disease. So how do we really study such an intricate system, and how gene expression is dysregulated in the context of disease? And in my work and now in my lab, we focus on the sequence-specific binding of transcription factors and their activity on enhancers and promoters, and how that leads to changes in gene expression. And so we predominantly, over the past 5 or so years, have been using ATAC-seg. So I'll introduce you to ATAC-seg, which is the assay for transposase accessible chromatin. And I'll give you a brief background on what we're able to do with that type of data, just so that everyone's up to speed before we dive in. So what can you do with ATAC-seg data? Well, first, the way that I like to think about ATAC-seg data is it's measuring where proteins bind to the DNA. So each of these proteins is a transcription factor, and ATAC-seg is essentially shining a flashlight on these transcription factors. And when you have one of these transcription factors bound in your actual ATAC-seg data, what you see is an increase in the signal, which we refer to as a peak. And this is indicating that there's a protein binding event here. Of course, if you lose one of those protein binding events, you will lose the corresponding signal in your data. And so we can use this, the existence of one of these peaks, to start to infer some things about the cell of interest. And so one of those things is we can ask, what genes might this protein binding event regulate? And that's, assuming that this is a regulatory element, either an enhancer or a promoter. And so we can do that based off of the proximity of a gene to that regulatory element. We can also scan the sequence underneath this peak and ask what protein might be binding here. This isn't ChIP-seq data, but we can infer what transcription factor might be binding based off of the sequence that we observe underneath that peak. And so in this example, maybe we find this GATA transcription factor binding site, and we say, okay, well, we think this is one of the GATA transcription factors. Of course, this is an inference, and we don't know exactly which GATA transcription factor is binding, because all of the GATA family factors bind approximately the same sequence. So we can do various analytical steps to try to predict which of these GATA transcription factors it might be, for example, using gene expression data from the same cells. And finding that only GATA2 is expressed, so we think that it's a GATA2 factor. But by and large, this is an inference, because we're not directly assaying which factor it is like you would in ChIP-seq. The other thing that you can do with ATAC-seg data, which I really won't talk about today, is transcription factor footprinting. But the use case that I'll really harp on quite a bit is this concept of allelic accessibility. And so, for example, if in this particular cell of interest, we had one allele that had a T and one allele that had a C, and if we thought that this was being bound by a GATA transcription factor, you would predict that the factor would only bind to the T allele and not to the C allele, because the C allele would no longer match this transcription factor motif. And so in the ATAC-seg data, what that would show up as is a predominance of the T allele in your sequencing reads compared to the C allele, which is not very strongly bound by transcription factors. So at a molecular level, how does ATAC-seg work? Well, when transcription factors bind to chromatin, they create these small nucleosome-free regions on either side of the transcription factor binding site. And those regions are what we refer to as accessible regions. And that's because when we incubate the chromatin with these transposase enzymes, the transposase can access those nucleosome-free regions. The transposase performs its normal duty as an enzyme. It performs a cut and paste reaction, and we are pasting in these little red and blue adapters, which simultaneously fragments the DNA and loads on these little primer handles that we can use for downstream amplification of these regions. And so you end up with a lot of these regions from accessible chromatin sites that were previously bound by transcription factors. And when we sequence those and align them back to the genome, they pile up in certain areas, and that's what we refer to as an ATAC-seg peak. So the last piece of information that I want to give you before we dive into this is that ATAC-seg data is extremely cell type specific. So here's a random locus that I've pulled out of our data. Each of the rows here represents a different cell type. So we have excitatory neurons, inhibitory neurons, microglia, and other glial cells. And I hope that you can appreciate that the diversity of chromatin accessibility just in this one locus is pretty astounding. So you have peaks that are highly specific to one subtype of neurons or another subtype or different types of glial cells throughout this locus. And this regulation of a single gene's expression, I think, really illustrates how cell type specific gene regulation is in general. And so we leverage that idea to try to understand disease, because the disease-specific gene regulation should be equally as specific. And so I'm going to focus the talk around a few questions, which I'll kind of paint on top of this diagram. The first being, how can we use single-cell approaches to better understand disease? And so I'll talk to you about our efforts using single-cell ATAC-seg, and I'll compare and contrast that to similar efforts with bulk ATAC-seg to show you really what the gain is here from single-cell assays. The second question I'll talk about is, how do non-coding sequence changes contribute to disease? And this will form probably the bulk of the talk. And at the very end, I'll touch on this question that I'm very interested in, and I think others are as well, about how regional and connective diversity in the brain is encoded and whether or not the epigenome plays any role in that process. So the first question, how can we use single-cell technologies to improve our understanding of disease? So the system of interest here that we're using is the brain. My laboratory uses primary brain samples from humans, so post-mortem frozen tissue from all of these different regions that we collect with the help of clinical collaborators. And so I'm going to show you some bulk ATAC-seg data from various different control individuals, healthy controls that do not have cognitive decline. And I'll show you data from each of these different brain regions, which, if you don't know what the brain regions are, it's not that important for the purposes of the talk, but suffice it to say that they are both cellularly and functionally quite divergent. So if we perform bulk ATAC-seg from each of these regions, there's about 100 or so samples on this plot. And I'm showing you a dimensionality reduction where each dot represents an individual sample, and they're colored by the region from which they were derived. And so I hope that you can appreciate that there's a few different clusters of samples here. For example, down here you have clusters from the caudate nucleus and the putamen, which are very spatially proximal and functionally proximal portions of the basal ganglia in the brain. And so their proximity down here makes sense. And then similarly, you have regions of the brain that are part of the isocortex or the hippocampus or the substantia nigra, which perform different functions, and therefore it's not surprising that they cluster separately. This is what we would expect, and it's largely driven by different cell types that only exist in the basal ganglia or in the isocortex or in the hippocampus. It's really a cell type driven phenotype that we're observing. And so you can observe that. So for example, in the striatum, if we look at our ATAC-seg data in the locus of the dopamine receptor D2, this is a receptor that's only expressed on neurons in these two regions, and you can see there's essentially no activity anywhere else. And you see the same thing for substantia nigra specific genes like IRX3 or isocortex specific genes like this voltage-gated potassium channel. So what I've shown you so far is that a lot of the heterogeneity that we're observing between these brain regions is driven by cell types that are specific to those specific regions. We started this whole effort quite a few years ago with the intention of using bulk ATAC-seg to understand disease processes. And we started by comparing control individuals that were cognitively normal, but some individuals had high disease-based pathology, so they might be on their way to developing Alzheimer's, and others had very low pathology. And when we compared those individuals, we ended up finding very few differences. And so I'm showing you here comparisons of different regions. And so when you compare different regions, you do see a lot of differences. The data is quite high quality and you can see differences when they exist. But when you compare individuals in that low pathology group to the high pathology group, there's essentially no statistically significant differences across any of these brain regions. And this was very surprising to us. There's the brains, visually under the microscope look very different. And we thought we would see huge differences in this comparison. And it turns out that the reason we don't see differences is because we're using these bulk profiling approaches, which obscure a lot of the signal in the data. And over the next couple of slides, I'll tell you about, I'll talk to you about the single cell approaches that we've used, and then compare and contrast those to the data derived from the bulk. So how do single cell approaches work? Well, we predominantly use the 10x Genomics Chromium system. This is a very easily adaptable system that gives relatively high quality data. And I do feel that pretty much any lab could do this. So that works by performing the transposition in bulk as if you were going to do bulk ATAC-seg, and then you use the 10x system to combine a single nucleus with a bar-coded bead, and that gets encapsulated in this oil immersion where each little aqueous droplet is a small reaction chamber. And this allows the primers that are present on the gel bead to amplify the DNA present in the nucleus. And so when you do this in these little reaction chambers, all of the molecules of DNA present in this particular nucleus here get barcoded with the pink barcode. These get barcoded with the purple, and these get barcoded with the blue barcode. And so now when you lyse open these aqueous droplets, any sequencing molecule with the purple barcode, you can map back to that same single cell. So you can, in a massively parallel reaction, you can do thousands of single cells and then sequence them all together and kind of computationally deconvolve which cell each read belonged to. So when we perform this in those same brain regions that I introduced you to before, we get something that looks like this. So this is a dimensionality reduction plot where each dot represents an individual single cell. The next challenge is to understand what all of these individual clusters that we call in our data actually correspond to. And normally if you were doing a gene expression based approach, you would look for which genes are expressed in which clusters, and you would define them based on gene expression. Because we're doing ATAC-seg, we're kind of one step removed from gene expression, but what we're able to do is infer gene activity or gene expression from the chromatin accessibility data. And I won't get too far into how that works, but suffice it to say that the local structure and activity of the chromatin serves as a very good predictor for gene expression. So for example, if we were to color each cell based off of the predicted gene expression for this VGLUT1 gene, which is highly specific for excitatory neurons, you would see that these clusters down here, which correspond to these clusters over here, make up all of the excitatory neurons. And we can do this for a diversity of markers, finding microglia, oligodendrocytes, astrocytes, OPCs, and also inhibitory neurons. And so this allows us to then group all of the inhibitory neurons together, or all of the excitatory neurons together, and create these cell type specific accessibility profiles that we would otherwise not be able to create because the brain is so heterogeneous. Okay, so what does the single cell data get us that the bulk data did not? So if we called all of the peaks that we could find from the bulk ATAC-seg data, we would get around 180,000 of those. In the single cell ATAC-seg data, if we call peaks in each individual cell type, we end up with around 350,000 peaks. And if we ask how many of the peaks from the single cell ATAC-seg data are overlapped by a peak in the bulk ATAC-seg data, it's a very small proportion, and most of the peaks in the bulk ATAC-seg data are overlapped by one of the single cell peaks. So what is causing this difference? Why are there so many more peaks in the single cell data? One of the reasons for this is that of the 350,000 peaks, half or more than half of those are actually cell type specific. There are, peaks, 50,000 or so peaks that are specific to only microglia, and you would only observe them in microglia. And so these peaks do have very characteristic transcription factor motif enrichments, transcription factors that we would expect to be heavily involved in the cell identity of excitatory neurons or in microglia such as SPI1 or BCL11A. And so these peaks make sense. We know that they are truly cell type specific peaks that have biological relevance. And so when we ask, what are the peaks that we're missing in our bulk ATAC-seq data, the peaks here that overlap the bulk versus the ones that are only in this gray box, we're finding much more peaks in the single cell ATAC-seq data that come from microglia, astrocytes, and OPCs, and these happen to be the more rare cell types. So when you have a cell type that is rare and you're doing a bulk assay, you're not going to recover the peaks from those cells. And so what I'll show you, so that's one reason why the bulk assays might not capture the disease relevant biology, especially for something where we think, maybe microglia are the important cell type of interest. The last bit that I'll show you is just how heterogeneous the bulk ATAC-seq samples are. And we're going to do this with an in silico deconvolution framework. So what do I mean by that? We're going to take the bulk ATAC-seq profiles that we got, and we're going to try to estimate what % of that bulk profile is coming from various different cell types. We're going to do this with this algorithm called CyberSort, which was developed by Aaron Newman at Stanford. And what this does is it defines regions that are highly specific to each cell type, like what I showed you in the previous slide. And then it uses that with a support vector regression to understand and predict what percentage of the signal might be coming from each of these different cell types. When we do this based off of the single cell ATAC-seq ground truth that we know, the prediction from CyberSort is extremely accurate when we're asking it to predict cell type contributions. And so we can look at all of our bulk ATAC-seq samples shown here along the x-axis and ask what % of each sample signal is coming from the various different cell types shown here at the bottom. And I hope that you can appreciate that there's a huge diversity even within a single brain region shown here by color along the bottom. So for example, if we look at the superior and middle temporal gyrus here in dark blue, some samples have extremely high percentages of oligodendrocytes, whereas others have up to 50 or so % of excitatory neurons. And so this creates a huge problem when you're trying to compare these samples. If the cellular heterogeneity is so strong, that will decrease your signal to noise ratio and make it very hard to find significant biology relevant to disease. The last bit that I want to talk about from a single cell standpoint is how useful single cell approaches can be to really tease out fine-grained cell states or cell types. So if we take all of the neurons from the previous clustering that I showed you, we can identify approximately 20 or 5 or 30 different subtypes of neurons. And so each of these groups, each of these colors is representing a different cluster. And for many of them, we can assign them to known neuronal cell types. And for some, we think that there's, kind of some novel biology peeking out here that we're still kind of investigating. And so, these occur in different proportions. We'll go over what all of these definitions are in the next slide, but suffice it to say that each of these clusters represents a unique neuronal cell type or cell state. And so if we can define these neurons based off of the inferred gene expression that I told you about before. So we can look at VGLUT1, which again marks excitatory neurons. We can look at various markers of inhibitory neurons like VGAT. And we can even drill down to extremely specific cell types. So for example, here I'm showing you DARP32, which is also called PPP1R1B. Which is a receptor that, or is a molecule that's expressed on medium spiny neurons in the striatum. And we see that there's 2 clusters here that are positive for this particular marker. And these turn out to be two different subtypes of medium spiny neurons, one of which projects to the substantia nigra and the other which projects to other regions of the brain. And so what this is showing us is that this single cell ATAC-seg data can differentiate between a single cell type, a single neuronal type in the brain that just happens to project to two different parts of the brain. But the neurons have a very similar function. Okay, so my last plug for single cell data is that we're actively developing software to analyze this data. If you're interested in doing single cell ATAC-seg or you're actively doing single cell ATAC-seg, I would encourage you to check it out. It'll be published in the next couple of weeks to month, but you can find it online. The software tool is called Archer, and it really facilitates the analysis of single cell ATAC-seg data for anyone, really, for people who aren't, bioinformatics researchers. I really feel that the ease of use of this tool is one of its strengths, and it allows you to do extremely complex analyses. Most of what I've shown you so far is just, a few commands in this software tool. So check it out if you're doing any single cell ATAC-seg. Okay, so with all that as a backdrop, how do non-coding sequence changes contribute to disease? So we're going to use some of the data that I've introduced you to understand this question. But before we dive in, I want to provide you with a little background on why this is such an important problem. So we're most interested now in Alzheimer's disease and Parkinson's disease. These are not somatic diseases for the most part. And the way that these have been studied at a genetic level is that we perform what are called genome-wide association studies, or GWAS. And what that is, you take thousands of cases and thousands of controls and you sequence those and you ask what genetic variants predispose someone, are significantly enriched in the cases compared to the controls. And so that would give you a table that looks something like this, where you have genetic variants highlighted here by their SNP IDs. You have the significance of their enrichment in the disease individuals compared to the controls. And oftentimes these tables will annotate a gene that they think is the causative gene in this association. More often than not, that is just the closest gene that was located to this SNP. And that's really where the problem begins. The SNPs that are identified are really tagging really large loci in the genome because of this propensity of nearby loci to be co-inherited. It's very difficult to say that it's this SNP and not the SNP that's one a few bases away because of this concept of linkage disequilibrium. I don't really want to dive into linkage disequilibrium in this talk, but suffice it to say that this SNP may or may not be the correct SNP, and it could be any SNP that's really located nearby and inherited together in the same fashion. In some cases, these genome-wide association studies have found the correct gene. So for example, ApoE we know is a clear driver of Alzheimer's disease. But there's many other loci here which we're not 100% sure what the driver gene is and even what the causative SNP might be. To make matters worse, most of the loci that are identified by genome-wide association studies have no potential coding effect. And so only a very small portion of all of these loci have a likely coding effect or a possible coding effect. And a much larger portion, we would say, are almost definitely or highly likely to exert a non-coding effect. And I'll talk to you a little bit about what we think those non-coding effects might be, but the underlying assumption is that they are not changing an amino acid sequence and they are not having a protein coding effect. So we think that they're altering the activity of the non-coding genome and gene regulation in general, likely by changing the sequence of a transcription factor binding site, much like what I showed you for that GATA transcription factor example in the very beginning of the talk. So genome-wide association studies are good at identifying large regions of the genome where genetic variation is associated with disease, but they're really not good at determining which cell type is affected, which gene is affected, and pinpointing which SNP is functional. And so we're going to start layering on a lot of multi-omic data to try to tease out what these non-coding loci might actually be doing in this disease. So we're going to use single-cell ATAC-seq to determine what cell type might be affected. We're going to use a combination of assays, including three-dimensional chromosome conformation assays, to predict which genes will be affected. And then we're going to use a machine learning framework to try to narrow down to the precise SNP that might be functional. So what cell types are generally affected by these diseases? We can use what's called LD score regression, which really just looks for enrichment of genome-wide association studies in the cell type specific peaks of various classes. So here I'm showing you the different cell types that we identified previously, and then a whole bunch of diseases here looking for enrichment of disease genetics in these different cell types. And so we're most interested in Alzheimer's and Parkinson's disease. We're able to recapitulate findings that have been shown before, where microglia are enriched for genetic variants that are associated with Alzheimer's. And we don't find any cell types that are enriched in Parkinson's disease, but we do find, a very strong neuronal enrichment in diseases like schizophrenia or neuroticism or ADHD. And importantly, when we look at kind of negative control diseases, we don't find any enrichment in neuronal cell types or brain cell types, because we wouldn't expect bone density or coronary artery disease or lean body mass to have an effect on the brain necessarily. We can subdivide down to even finer definitions of cell types, but we still don't really see an enrichment in Alzheimer's or Parkinson's for any of these neuronal cell classes. There's various reasons why that might be. I'm happy to talk about it in the Q&A at the end. Okay, so we think that microglia are an important cell type in Alzheimer's, but this is still a very generalized approach. We really care about single loci and what genes and cell types they affect. So to do that, as I mentioned, we use chromosome conformation data. We're particularly using HiChip, which uses an antibody, in our case, to the active histone-marked H3K27 acetylation, which allows us to enrich for interactions between enhancers and other loci. This really opens up the technology to primary samples. So we're using primary frozen human brain here. And so without this, it would be quite difficult to obtain the cell numbers to do this. The other method that we can do to associate, a gene promoter with a target enhancer would be using co-accessibility. So what do I mean by that? Well, if we have a promoter and a couple of different enhancers nearby, we can use our single-cell ATAC-seq data to identify when this promoter is accessible, which of these other enhancers is also accessible. And so, here's just some toy data where each row represents a different cell and they're grouped by different cell types. And I hope you can appreciate that when the promoter is accessible, this enhancer is often accessible as well. And so we could represent this as a correlation or as a heat map. And the real trick here is just trying to associate promoters and enhancers based off of their co-accessibility across a large number of cell types. So when we do this HiChip and co-accessibility, how many genes can we putatively associate with these various diseases? So if you just took the tables from the genome-wide association studies and said, okay, the nearest gene is the causative gene in that disease, for Alzheimer's disease, you would end up with around 51 genes that have a potential involvement in Alzheimer's. When we expand this kind of universe of genes that is potentially involved, we can potentially link 400 genes through HiChip and single-cell ATAC-seq, really expanding this haystack, which we're eventually going to whittle back down. You can see a similar trend in Parkinson's disease, where you have many more genes linked through this HiChip and single-cell ATAC-seq framework. Okay, so I've introduced you to all of the data that we're going to use. How are we going to drill down to these cell types, genes, and functional SNPs? Imagine you start with a single entry in that genome-wide association study table, so you have your lead SNP. We expand that SNP to include all of the SNPs that are co-inherited through linkage disequilibrium. And so this represents all of the SNPs that we think could be driving this disease association. And we're going to slowly remove SNPs based off of certain filtering criteria. So the first one is we think that these SNPs are disrupting or causing disease based off of disrupting transcription factor binding. And so we're going to limit them to only the SNPs that overlap with peaks in our data. We're then going to predict which SNPs have strong effects, so looking at which SNPs alter sequence binding motifs. So here we would predict that this C to A change is going to disrupt this transcription factor motif. And then once we've nominated an important potentially functional SNP, we're going to try to link it to various genes using high chip and single cell ATAC-seg. The crux to all of this is predicting these SNP effects, which we do with a machine learning framework. So what we do is we take all of our individual clusters and we make a machine learning model for each cluster where we provide that model with about 60,000 regions that are accessible and about 60,000 matched regions that are not accessible. And we're essentially teaching the computer to understand the underlying grammar of accessibility and be able to predict on a per base level how accessible a single site might be. So then we would take our wild type and variant sequences derived from each of these potentially functional SNPs, feed it to that algorithm and ask it to predict the sequence-specific accessibility for disease and non-diseased alleles. And what we would hope to get out of this are these kind of little logograms like this, where the larger the letter is, the more accessible it's predicted to be. And we can subtract and create kind of a delta profile, which would show you that the effect allele here is predicted to be more accessible than the non-effect allele. And we would hope that we could find a transcription factor binding motif within this delta logogram. So how does all of this come together? I'll walk you through a few examples which will show you each of these different steps for a couple of different loci that we think are super interesting. So the first is the PICALM locus. Here you have the lead SNPs identified by genome-wide association studies. If we expand that in linkage disequilibrium, we end up with about 165 SNPs, 24 of which overlap peak regions from our chromatin accessibility data. When we look at the nearby genes, most of these SNPs surround this gene PICALM, which is why the genome-wide association study has assigned this as the active gene. And I'm going to try to convince you that it's this SNP right here, which is a few kb upstream of PICALM, that's actually exerting the functional effect in this locus. So when we look at our single cell ATAC-seg data, again, we're looking at the peak regions here indicating transcription factor binding. This particular SNP overlaps a very oligodendrocyte-specific peak, predicting that this locus would be functioning through oligodendrocytes. We can ask what genes is this SNP linked to, and we can see that it through high chip communicates with the PICALM promoter and also the promoter of this gene EED, which is a polycone protein. And we can see the same linkage here with the PICALM gene through our co-accessibility framework. But so why did I pick this particular SNP? Well, when we look at the actual sequence in our machine learning framework, the A allele here, which is the effect allele, has very low accessibility. The G allele has much higher predicted accessibility. And when you take the delta of those, you can hopefully see that this matches almost perfectly to a FOS motif, indicating that this FOS family transcription factor is potentially disrupted in its binding based off of this particular single base change. This in and of itself isn't, full functional evidence that there's a bias in transcription factor binding. But if we go back to our bulk ATAC-seg data, which we now have from hundreds of patients, we can ask, do we see any allelic accessibility at this particular site? And so what we're looking for here is reference alleles and variant alleles, where each dot represents a specific sample. And hopefully you can see that the reference allele, which is the non-effect allele here, is much more accessible than the variant allele. And this provides quite strong evidence for the fact that there is a difference in transcription factor binding at this particular site. So I'll walk you through a few more examples of how this all looks. So here's another locus, the KCNIP3 locus. Here's the lead SNP over here. When we expand in linkage disequilibrium, there's about 100 SNPs, 20 or so overlap peak regions. The locus looks like this, and I'm going to kind of highlight these two genes, KCNIP3 and MAL. There's two SNPs that we think could be functional, and they lead to very different interpretations of this locus. This SNP down here overlaps an oligodendrocyte-specific regulatory region, and this SNP over here overlaps a neuron-specific regulatory region. The different loci talk to different genes. We find that this SNP seems to be talking most strongly to the MAL promoter, and the KCNIP3 SNP is talking to the KCNIP3 promoter here. And they have support in different ways. So the MAL SNP, which is an oligodendrocyte-specific SNP, the machine learning algorithm predicts that it would have a very strong effect on accessibility, especially in the context of this SOX motif. So changing this T here to a G ends up destroying this SOX motif. And we know that SOX factors are hugely important for oligodendrocytes. Unfortunately, in our bulk ATAC-seg data, we didn't have any heterozygotes at this locus. In the other locus, the KCNIP3 locus, we do see this allelic accessibility that I introduced you to before, where the reference allele is much more strongly accessible than the variant allele, but our machine learning algorithm does not pick up a motif that's being disrupted. And so we're still trying to tease out exactly which of these might be the case. It's possible that they're both correct and there's two effects of this locus. But this is just illustrating for you the complexity of how all of this works. So just to kind of breeze through one last example, here is the BIN1 locus. There's a few SNPs that we think might be important, in particular this leftmost SNP here. It overlaps a very strong microglial specific peak. It communicates with the promoter of the BIN1 gene, and our machine learning algorithm predicts that this C to T change disrupts a KLF4 motif, which is a very specific microglial transcription factor. And again, we don't have any support from our bulk ATAC-seg data for this locus. So if you're interested in the brain, these three loci that I've shown you, they might not ring a bell. They might not be super well known to you because, you know, that's the point of what we're doing. We're trying to introduce new genes that might be involved in these diseases. But I'll, in the last couple of minutes, go through an example of a very well-studied locus and show you what we can do with all of this multi-omic epigenetic data. So I'll introduce you to the MAPT locus. This gene, MAPT, encodes for tau protein, which is a huge player in Alzheimer's disease in terms of its role in neurofibrillary tangles. But it also is one of the strongest genome-wide association study loci that's associated with Parkinson's disease, which is not a disease that we think of you know, having a role with tau or neurofibrillary tangles. It also happens to be extremely interesting from an evolutionary standpoint. So there's two haplotypes of the MAPT locus that differ based off of an inversion. So this breakpoint here and this breakpoint here are actually flipped in the different haplotypes. So the H1 haplotype is in this order, the H2 haplotype is reversed. And then in addition to that inversion, there's over 2,000 SNPs that differ between the H1 and H2 haplotype. And so when we look at gene expression data across many individuals, you can see that the H1 haplotype has higher expression of MAPT, the H2 haplotype has lower expression of MAPT. And so our question was, what are the epigenetic differences in these haplotypes that are driving this change in gene expression? And so the way that we're going to do this is using an allelic comparison of these different samples. We, of course, have individuals that are homozygous for H1 or homozygous for H2, and then we have heterozygotes. For the homozygotes, it's very easy. We align to a genome that has been masked with N bases so that these SNPs can't play a role in differential alignment. And then we take the H1 and H2 reads and we do differential ATAC-seg or differential high chip in those settings. For the H1 and H2 individuals, we will split reads based on whether they have one of these SNPs for the H1 or H2 haplotype to get H1 specific or H2 specific reads and then perform differential analysis. And this comparison is actually really nice because it's internally controlled, right? If you have a heterozygote and you take the H1 and the H2 reads, you don't even need to perform normalization because they're coming from the same cells. So here's that locus again. I'm showing you this within the confines of the two breakpoints. If we look at the ATAC-seg data from H1 and H2 individuals, we can identify a few regions. This peak here is stronger in the H2 than the H1, as are these. And then this peak over here is essentially completely absent from the H2 individuals. And so we're going to anchor our analysis on some of these ATAC-seg peaks to try to understand how they're different in these two haplotypes. So if we take our high chip data and we ask at this centralized point here indicated by the dotted line, what is the propensity of the rest of this locus to interact with that dotted line? That's shown here along the y-axis, and the two different haplotypes are shown in blue and red. I hope you can appreciate that there's this huge peak of interaction coming from here. So there's a genomic interaction occurring between these 2 points. And we're going to label those A and B and follow those throughout this assay. So we can kind of march this dotted line over to this other peak, which is the MAPT promoter. And you can see an increased interaction here on the shoulder, which is overlapping this MAPT H1 haplotype specific peak. And there's a bit increased interaction downstream that you can't quite see here. So if we keep marching this down, now you can see that increased interaction coming from this enhancer with the MAPT promoter. And if we go all the way, you see the reciprocal interaction that you saw here with points A and B. So that's all well and good. This explains some of the gene expression differences that we see in this locus, where there's some genes here at point A, which are much more strongly expressed in the H2 haplotype, or at point B. We don't really think that these are driving the disease biology. They are non-coding RNAs or pseudogenes that don't have a very strong annotated function, but we're still looking into it. But the real question is what happens outside of the MAPT locus, because if you remember, this is inverted. So the orientation of what's in the middle is changed with respect to what's outside. So now we'll zoom out. I'm showing you those same inversion breakpoints here. And if we look at the MAPT promoter, I hope you can appreciate that both from homozygotes and from heterozygotes, we see an increased interaction with this potential regulatory element upstream. We can zoom in on that regulatory element and find some neuronal specific peaks. This is consistent with the role of MAPT being a neuronal specific gene. And so we come up at the end with this model where the H1 haplotype, points A and B are not interacting, and this distal enhancer here seems to, or potential enhancer, seems to be interacting with increased frequency with the MAPT gene, which would lead to a higher overall MAPT expression. And the H2 haplotype, points A and B interact very strongly, which seems to insulate the MAPT gene from this distal enhancer downstream. I'll just briefly say that we're following up on a lot of this work using massively parallel reporter assays, which assay the differential activity of different SNPs, and also with scarless base editing using either CRISPR tagged with a deaminase or Cas9 tagged with a reverse transcriptase, which is prime editing from David Liu's lab. And we're trying to generate iPSCs that differ by a single base and then differentiate them into cell types of interest and demonstrate the cell type specificity of our predictions. So I'm going to skip over the last part of the talk out of the timing. Sorry, I can't see this anymore. And just give you the summary. So hopefully you've seen today that tissues and cell types are complicated and multifactorial. We can identify, up to 30 different neuronal populations in our data. The chromatin accessibility landscapes of these cells are extremely cell type specific. More than half of the peaks that we observe are specific to one or a subset of different cell types. The vertical integration that I've shown you of multi-omic data can be really powerful, and this is only going to improve as our analytical methods improve, and it can really provide some key insights into loci that have remained a little enigmatic over the years. And if you take home nothing else from this talk, hopefully you appreciate that the non-coding genome really does matter, and diseases are more than just collections of coding mutations. So with that, my lab is really interested in all of these different approaches to studying epigenomics. And I'm really thankful to have the opportunity to work at such an amazing place as the Gladstone Institutes and UCSF. We're definitely recruiting, so if you're interested, let me know, and I'll just thank the various people who have been involved in the work and the agencies who have funded my research over the years. So with that, I'm happy to take any questions.
There's one question in the chat. Do you want me to read it, or you want to read it?
I can read it. So the question is, the ATAC-seg peaks always represent that the corresponding gene, or I guess, do the ATAC-seg peaks always represent that the corresponding gene is transcribed? Since there are enhancers that are open but not necessarily active, so how does that all work? How might this affect the interpretation of the differences at the single cell level? Maybe I'll start answering some of these so I don't get too far ahead of myself. So the correlation of chromatin accessibility and expression is not necessarily one-to-one. So you can have high chromatin accessibility and it won't change expression at all. This depends in large part on the gene. Some genes are very regulated by enhancers, others are less regulated by enhancers. So for example, we find that genes that are involved in kind of the immediate immune response, those often aren't regulated at the level of an enhancer. My hypothesis is that the cell wants to express those really quickly in response to a stimulus, and so having to turn on a new enhancer and do all these things may or may not be the most efficient way, and I think that they use more of a kind of paused polymerase method of immediate expression. So there's not really a one-to-one correlation. Certainly understanding what ATAC-seg peaks are important for the expression of a gene can be resolved by high chip, which is suggested in the question. And then the last part of the question is, can you do ATAC-seg, high chip, and RNA-seq from the same single cell? Currently, that's not a possibility, but I'm sure, well, at least not in a commercial platform. There are many people who are interested in those types of assays and developing them, but they have not been widely adopted. But it is much more common to do ATAC-seg and RNA-seq in the same single cell now that that has been commercialized by 10x Genomics.
I don't know if there are other questions coming by through Alfredo. Let's wait. It takes a few minutes before everything moves.
No problem.
I'll ask maybe a very, 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 very stupid question. This is a huge amount of work for every single gene that you identify. How long does it take you to do all the deconvolution in the computers and everything?
Yeah, it is a very time-intensive process, and so far, the analytical methods are still catching up, and they're relatively customized as well. So if you have a locus that you're interested in, analytically dissecting it can take a week or two to really convince yourself that what you're looking at is real. And so if you think about Alzheimer's disease, there's at this point probably 70 or so loci that are implicated, and you wanted to do that one person, it would take you years and years to go through all of this. And so in some ways that's why we use this layered approach, because we have to go through all of these loci in a much more expedited fashion. And so the machine learning and all of that kind of expedites the hands-on time that we have to interact with the data. So it can take a really long time, and that's why these analysis methods are trying to create very clear filters where you can eliminate different SNPs at different stages of the analysis. Will we get to a point where this is kind of a one-size-fits-all solution and you can take any disease you're interested in, just hit a button and it goes? I don't know if we will get there soon, but I'm sure we will get there, and certainly the analytical portion of the field is kind of moving in that direction. So I don't know if that fully answers your question. From a hands-on standpoint, it does take time, but we're really trying to automate all of this so that there's not as much human input needed in the process.
Okay, there's a question in the chat from Diego Arturo.
So many of the functions of the brain are carried out by groups of neurons in engrams. This group of neurons are activated and highly plastic to convey a specific task. Would it be possible to extract specific info from accessibility from an engram point of view? This is something that we're super interested in. So engrams are kind of, the encoding of a memory or a specific response. And there are a lot of groups who have painstakingly made genetic models which will label specific neurons that are involved in an engram. And there have been a few recent publications where they've isolated out those labeled engram cells and specifically performed single-cell RNA-seq. No one has done single-cell epigenetic profiling yet to try to identify what's different about those engram cells compared to other non-engram cells. It's a super interesting field to try to really understand what is a memory, how is that encoded, and I'm very interested to see how the epigenome plays a role in that. It's something that we're not actively working on, but super interested in if anyone is interested in collaborating. There's another question here. The peaks called by single-cell ATAC-seg and not by bulk ATAC-seg are most likely from highly cell type specific peaks. What are the peaks called only by bulk ATAC-seg and not by single cell ATAC-seg? Could they be false positives? So I think you're exactly right. I do think that they are false positives and that results from the peak calling algorithms, not necessarily from the data being poor quality, but there is just some aspect of peak calling that provides false positives. And so if we had used maybe a more restrictive threshold of what a bona fide peak would be, we might not see those. It's about 6 or 7% of the total bulk ATAC-seg peaks aren't observed in the single cell ATAC-seg data. And so I think that you're right. I do think that they're false positives.
Eddie goes on and makes another question.
Yeah. In a previous seminar, we were told that more than 90% of the GWAS loci were in non-coding regions, but here at least 17% is likely coding. Do you think that this proportion of coding and non-coding regions could also be a cell or context specific? Are there previous reports or evidence for this? So the percentage of loci that are non-coding, it varies from disease to disease. I would say that it's somewhere between 75 and 90%, and some of that depends on how you categorize the loci. So in the plot that I showed you, the likely coding loci is basically any locus where one of the linkage disequilibrium SNPs affects an amino acid, even if that amino acid change is predicted to be a benign change. So we've used a very stringent threshold for a likely non-coding locus. If you were to, only take loci where potentially detrimental amino acid sequence changes are used, then you would probably get much closer to that 90% mark. But as you suggested, definitely depends on the disease that you're interested in, and also probably how many GWAS loci have been found for that disease. Another question from Victor. You talk about cell type specificity. How dynamic do you think chromatin accessibility is in processes like memory consolidation and recall? I wish I knew the answer to that. It's a great question. I don't know if that's been done in the field. I haven't seen it if it has, but my lab doesn't precisely study memory. I would expect that to the degree that transcription changes in the process of memory consolidation, the epigenome will also change. So I guess I base my interpretation on the transcriptional work that's already been done, which does show some transcriptional changes, but they do seem to be relatively small, at least in my opinion. So yes, I do think that chromatin accessibility will change in the process of memory consolidation, but probably only to the extent that it's involved in changing transcription. Another question from Martha. How well does ATAC-seg work for chromatin remodelers and for in situ and transient transcription factor modifiers such as kinases, simulators, etc.? I'm not sure I know the answer to that question. Certainly, things like the BAF complex, we don't, I don't personally think that the ATAC-seg does a really great job of capturing the activity of the BAF complex. And the more transient an interaction is with chromatin, with the chromatin, the accessibility will be correspondingly affected. And so I personally think that what we observe with chromatin accessibility is the binding of transcription factors that have relatively strong binding or relatively long binding and kind of similar on and off rates where they bind to the chromatin and they might come off the chromatin, but they bind again immediately. So I think these transient factors, you can pick them up if they're binding across every single cell. But if the binding is very heterogeneous, you might not pick them up with ATAC-seg.
Emiliano.
I wonder if there would be an individual cluster of peaks for samples coming from individuals with, for example, autism spectrum disorder or similar disorders, which are typically considered developmental disorders. So there's a few groups that are doing this. Will Greenleaf's group just had a paper that came out on bioRxiv about trying to understand the developmental brain from a chromatin accessibility standpoint. And I do think that if assayed at the correct time point, you would find peaks of chromatin accessibility that are specific to individuals with autism spectrum disorder or other neurodevelopmental disorders. I think the challenge becomes understanding how heterogeneous that disease is. So is there only one way to develop autism, or are there 100 different ways to develop autism? And if there are 100, then it becomes much more difficult to identify changes that are specific to autism because there's many different ways to develop the disease. So I do think that there will be differences. I mean, the cells are behaving differently. I think that the underlying epigenome will change, but really teasing out those differences will require a substantial amount of work. Question from Diego, am I aiming to use optogenetics in the future? So my laboratory, I'm very interested in using optogenetics. I think, you know, my laboratory has at its kind of beginning been focused on human-centric assays. One of the reasons why I came to the Gladstone Institute is because there's so many amazing researchers here that have really amazing mouse models, some of whom are using optogenetics to control cell state and cell activity. And so there's a lot of interesting questions that we're hoping to explore with collaborators here at Gladstone, trying to understand, you know, how various different optogenetic stimulations affect chromatin and affect gene regulatory pathways. So I would say, yes, we're super interested in that, but we haven't gotten into it yet. Follow up to Victor's question. Yeah, so this is just kind of helping me answer a previous question. So there apparently have been new models in memory that show that the remodeling of TAD clusters is involved in consolidation. I think that's probably coming from Jennifer Phillips-Kremen's lab, if I'm remembering correctly. Another question here, what is the relevance of looking at the chromatin accessibility in complex cognitive processes other than correlations? Oh, I lost that. Is there any functional, is there any proved functional consequence at the circuit level? I wish I knew the answer to that question. I don't know of anyone who is combining kind of systems level neuroscience with epigenetics. Certainly, I would imagine that there are some circuit level differences in chromatin accessibility. We're very interested in understanding this connective diversity of neurons and how that's encoded. So I wish I knew. I don't know the answer. Hopefully someone out there is combining that sort of systems level neuroscience with epigenetics. Another question, did we identify any clusters that are segregated by sex because the prevalence of neurodegenerative diseases is different across different sexes? Do we think that there are any biological differences, and can we use that information for diagnostics? So we weren't necessarily powered to identify differences in sex at the single cell level. From the bulk ATAC-seq level, there's very few differences based on sex, but then again, there's very few differences based off of that high pathology, low pathology, so it could just be a heterogeneity problem. We're actively trying to increase the N that we have on the single cell assays, and I imagine we will find differences that are based on sex, and I do think that those can be used in the future to try to understand why different sexes have different predispositions to these diseases and how we can use that in the clinic. I think that's the last question that I see here.
So maybe I'll do another question. Do you know if there is any epigenomic study that has already been done in the COVID patients that have neurological sequels to the disease?
I don't know if that has been done. Certainly it's being done for some of the other organ systems, you know, heart and other cardiac related symptoms from COVID. You know, the problem with the brain is we can't get it till the person has died. So, and for good reason. So I don't know if those studies have been done yet. I'm sure they're in progress. We don't have any COVID related work going on. We don't have any collaborators in that space, but if anyone's interested, we would certainly be interested in understanding that.
All right. All right. Well, if there are no more questions, I think we can only thank you for this crystal clear but very complicated seminar for those who are not totally familiar with that, but it was really very, very, very interesting, and I think very illustrative of what is being done nowadays in terms of dissecting all those diseases nowadays.
Well, I really appreciate the opportunity to speak, and please feel free to reach out to me if you have any other questions. I'm always happy to chat.
I'm sure some of our students will reach out to you and other people as well.
Great. Thanks so much.
Thank you so much.
Thank you, Ryan.
Thank you, Ryan.
You're welcome. My pleasure.